Welcome back to the deep dive. Today, we're taking a very necessary and serious look at the rapid evolution of prostate cancer research. This field is moving at warp speed, and our mission here is really to cut through the noise and synthesize the most critical recent updates.
That's right. We're looking at a stack of very current data, mostly from late 2025. And what we're seeing isn't just, small steps. It's a full-scale assault on the disease. We're talking complex, multi-layered clinical trials and these powerful new AI platforms that are designing the next wave of drugs.
Right. And, traditionally we might start with the AI that guides the work, but today we're actually going to flip the script a bit. We want to focus on the immediate human impact. So we're starting in the clinic with the phase 2 and 3 trials. These are trials actively testing combination strategies. They're designed to overcome drug resistance and, just as importantly, deliver better quality of life.
And that's the key. For you, the listener, the goal is to understand not just what drugs are being tested, but the really sophisticated biological thinking behind these new combinations.
Okay, let's unpack this. We're starting where the rubber meets the road, advanced clinical trials. So when we look at metastatic disease, the cancer has often, evolved to resist single drugs. Which brings us to a fascinating phase 2 study, a triple pathway attack using 3 investigational agents that hit the cancer from completely complementary angles.
This triplet therapy is it's really a masterclass in strategic combination. The 3 agents are SHR-3680, which is Rezvilutamide. That's a hormonal agent. Then you have HS-2009-3, an antibody-drug conjugate, or ADC. And finally, SHR-2554, which is an EZH2 inhibitor.
Okay, that is a lot of moving parts. To really grasp this, we need to understand the mechanism here. I mean, why layer an ADC and an EZH2 inhibitor onto what is essentially a standard hormonal backbone?
It's a great question. The primary driver of prostate cancer is the androgen receptor pathway. That's what SHR-3680 targets. But here's the critical vulnerability they're exploiting. We now know that androgen deprivation therapy, the hormonal treatment itself, actually indirectly enhances the expression of a surface protein. It's called B7H3.
Wait, so the therapy itself is creating the target for the second drug? That sounds incredibly intentional.
Precisely. You can think of the hormonal therapy as turning on a new beacon, this B7H3 on the cancer cells.
Right.
And that's where the ADC, the HS-2009-3, comes in. It's essentially a guided missile. The antibody part finds that newly visible B7H3 beacon, and the drug conjugate delivers its toxic payload right into the cell.
So the hormonal agent makes the cancer visible, and the ADC takes it out. Okay. Where does the third agent, the EZH2 inhibitor, fit into this layered attack?
That's the epigenetic insurance policy, or you could call it the shield breaker. EZH2 is a key epigenetic regulator. Cancer cells are just masters of adaptation. They use these epigenetic changes to develop aggressive treatment-resistant forms, especially after you hit them with hormonal therapy. So inhibiting EZH2 disrupts those mechanisms. The goal is to restore the tumor's sensitivity to the first 2 attacks and basically stop it from evolving new defenses.
That rationale is incredibly compelling. But, I mean, combining 3 powerful agents must raise concerns about toxicity, right? Especially for patients already dealing with advanced disease.
That is always the primary concern in phase 2, and it's why the endpoints are so closely monitored. The hope, though, is that by making the attack so targeted and so comprehensive, you can actually manage the overall effective dose. The primary endpoints for this trial, they reflect that. They're looking for things like durable PSA responses, including the rate of undetectable PSA at 6 months.
Which is a very strong indicator of long-term control.
A very strong indicator, yes.
Now, another phase 2 combination strategy tries to wake up the patient's own immune system. This brings us to the evolution trial. It's combining the established radioligand, 177-Lu PSMA 617, with dual checkpoint inhibitors, ipilimumab and nivolumab.
Right, and this targets a fundamental challenge. Prostate cancer is often considered an immune cold tumor. What that means is the immune system rarely recognizes it or attacks it effectively. Checkpoint inhibitors, when used alone, have really underperformed here.
So why pair them with a targeted radiation agent like 177-Lu PSMA 617? What's the thinking there?
The key is that this kind of targeted radiation, it doesn't just kill the cell. It often induces what's called immunogenic cell death. So when the cell dies in this specific way, it releases these alarm signals, these DAMPs, that basically flag the dead tumor cell to the immune system.
So the radioligand acts as the immune system's spark plug.
Exactly. The radiation exposes the cancer, and then the checkpoint inhibitors, ipilimumab and nivolumab, they step in. They remove the brakes the tumor uses to hide from that amplified immune response. You're looking for a synergistic effect, turning a cold tumor hot.
And the early results from this trial, which enrolled 93 patients in Australia, they suggest this strategy is working.
They do. The early data showed that 33% of patients on the combination were free of progression at 12 months. That's compared to just 17% on the radioligand alone.
Wow, so you're basically doubling the progression-free rate at 1 year.
In this patient population, that's a very significant signal. It suggests this could be the breakthrough needed to make immunotherapy viable in advanced prostate cancer.
So moving up to phase 3, we have a trial that focuses not just on survival, but really on quality of life. This is for men getting first-line treatment for metastatic hormone-sensitive prostate cancer. It's a direct head-to-head comparison of two next-gen hormonal therapies, Rezvilutamide versus the current standard, Enzalutamide.
And this trial is addressing a very real-world problem. Enzalutamide works, it improves survival, but it has a big downside. It has a known profile of neurologic side effects, fatigue, dizziness, sometimes even seizures, and that's linked to the drug easily crossing the blood-brain barrier.
Okay, so the core value proposition for Rezvilutamide is a smarter drug design that limits that brain penetration.
That is the critical difference. Rezvilutamide is specifically designed to minimize penetration into the brain. If it can maintain the same level of efficacy as enzalutamide while significantly cutting down on these debilitating side effects, it could. Well, it could rapidly become the new standard of care.
Which means the success here hinges not just on survival, but crucially on patient-reported quality of life. It's a really important shift, recognizing that living longer is only half the battle.
It is a very important one.
Okay, we've looked at complex combinations in advanced trials. Let's take a step back now to the absolute cutting-edge new agents just entering human testing. We're moving to a phase 1 trial in Australia for RAD402, a novel radioligand therapeutic.
What's fascinating here is both the target and the payload. The antibody, RAD402, targets KLK3. That's the gene that encodes for prostate-specific antigen, or PSA. Since PSA is so abundant in tumors, it's a very clean way to get the drug right where it needs to go.
But I'm really intrigued by the payload choice here. Terbium-161, we hear a lot about lutetium-177, which is a standard beta emitter. Why is Terbium-161 the focus for a brand new phase 1 candidate?
This is a breakthrough in isotope physics that could solve a major treatment challenge. Standard radioligands, like lutetium-177, they primarily emit medium-energy beta particles. They're effective, but they have a range of several millimeters. So they kill the target cell, but also radiate some healthy tissue nearby.
And Terbium-161 changes that radiation profile.
It does. It has a dual emission profile. It delivers those beta particles, yes, but it also delivers a significant punch of low-energy Auger electrons. And these electrons have an incredibly short range. We're talking nanometer micrometer distances.
So if the cancer cell is a few micrometers wide, the entire lethal dose gets delivered right inside the cell or immediately next to it with almost no scatter.
Exactly. It's a microscopic scalpel. It concentrates that lethal dose intensely over a tiny area. This dual emission profile may be particularly advantageous against micrometastases and isolated cancer cells, the tiny, hard-to-hit pockets of disease that often cause a recurrence.
So if it can spare surrounding tissue more effectively while delivering a more localized lethal punch, that could be a huge deal. It makes its progress through phase 1 absolutely essential to watch.
It really does.
It's clear that all this clinical innovation depends on highly specific biological rationales. And that specificity is being driven by a computational revolution, which brings us to a framework called DeepTarget. It feels like this tool is designed to address a scientific relic.
It does. For decades, the model in drug discovery was lock and key. One drug, one target. The scientific community sort of clung to that single target idea because it made screening and regulatory approval simpler. You know, it gave you a clear mechanism of action statement.
But the reality, especially in complex disease like cancer, is so much messier.
Absolutely. DeepTarget recognizes that modern small molecules are polypharmacological. They rarely act through a single isolated target. Its breakthrough is mapping both the direct and indirect anti-cancer mechanisms across all these different cellular contexts. It provides a true systems biology view.
So how does it manage that enormous complexity? What's the secret sauce?
It integrates three massive complementary data sets. First, you have large-scale drug viability screens, basically how well drugs kill cells. Second, genome-wide CRISPR knockout data, what happens when you delete specific genes. And third, matched omics profiles, which give you the whole biochemical context of the cells.
So rather than just modeling physical binding, DeepTarget is modeling functional behavior.
Precisely. The logic is simple, but so powerful. If knocking out a gene with CRISPR makes a cell react in the exact same way as giving it a drug, then that drug is very likely interfering with the pathway controlled by that gene. The relationship is established by function, not just structure.
That immediately speeds things up, especially for drug repurposing. You could find a compound approved for one thing has this unknown indirect effect that makes it powerful against cancer.
It truly opens up the existing therapeutic arsenal. DeepTarget uses a huge foundation from DepMap, the cancer dependency map, which has data on 1,450 drugs across 371 cancer cell lines. Its performance is measured with a metric called the Drug KO Similarity Score, or DKS.
Could you simplify what that DKS score is really measuring?
The DKS score is essentially a measure of functional mimicry. It asks, how closely does the cell's response to the drug mimic its response to knocking out gene X? A high score means the drug functionally looks exactly like a knockout of that gene, and the data proves this model works. DeepTarget outperformed recent structure-oriented tools in 7 out of 8 benchmarks.
If we connect this back to that triplet therapy we started with, the hormonal agent, the ADC, and the EZH2 inhibitor, it seems like tools like DeepTarget could optimize those combinations even further.
Oh, absolutely. The clinicians are using complex biological rationale now, but they're limited by known targets. DeepTarget can find hidden secondary targets that might be causing a small percentage of resistance. Identifying those could let us add a 4th layer of attack, or maybe substitute an existing agent with one that has a better polypharmacological effect that DeepTarget just uncovered.
This has been an incredible deep dive, really showcasing the multi-layered offensive against prostate cancer. We've seen the sheer complexity of clinical trials layering, hormonal agents, ADCs, epigenetic inhibitors, immunotherapy, all while launching phase 1 trials for next-gen radioligands like Terbium-161.
And if we connect this to the bigger picture, the number of phase 2 and 3 trials launching at the same time, all based on this increasingly sophisticated rationale from tools like DeepTarget, it shows a fundamental shift. We're moving away from just treating symptoms and toward treating the complex, evolved biology of the tumor itself.
And this brings us to a final, provocative thought for you to consider. If AI platforms like DeepTarget can so effectively map the multi-target approach needed to defeat complex diseases like cancer, are we rapidly entering an era where single-agent therapies, except maybe for early-stage disease, become ethically questionable given the proven efficacy of these sophisticated multi-layered attack strategies?
The computational power is outpacing the clinical capacity to test all the possible combinations. It's a truly dynamic race.
Stay strong and fight on.